All right, time to go teach. Hello and welcome to this next episode of Tutor Tutors, continuing in our unit with the cell, looking at the different characteristics of life that a cell exhibits. And in this case, we're going to actually look at the actual life cycle of a cell, thinking about how a cell goes about and reproduces. So here we go, our targets of the day. First is to describe the actual life cycle of a cell. That is our goal. Next is to recognize the different phases of the cell cycle. There are different moments in a cell's life that are actually going to be very critical for the process of it reproducing. And then lastly, we are going to explain key events that are going to take place during each of those phases. What are the main things that happen as the cell grows and develops? So, again, growing and developing, all cells are going to grow and develop, and pretty much all of life has this pre-reproductive stage and a ability to reproduce stage. And in some cases, we would call that reproductive stage the adult stage, but the key is that life starts off for an organism, it is unable to reproduce, and then it grows and develops to the point where it then can actually reproduce. And that works for a cell as well. And we can think about this in many different organisms. You could have a chicken where it has a life cycle where you have a chicken, which then lays an egg, which then hatches to be a chick, which grows up to be a chicken, which will lay an egg. And this goes on and on and on. And then that has the old question, which is, which came first, the chicken or the egg? And, your guess is as good as mine. We also have this with frogs, where the frogs are going to lay eggs. The eggs develop into tadpoles, and the tadpoles are going to develop into the next stage of the frog, and then we have this whole cycle reproducing again and again. And it is the same thing for humans. Humans have a child, which can grow up to then have another child, which can grow up to have another. And so this process continues and continues. And that is the life cycle for these different types of organisms. For a cell, we call this cycle the cell cycle. So all of that to bring us to the fact that those other organisms that I actually showed, the chicken, the frog, the humans, they all reproduced sexually. And so even though I only showed one individual, obviously one individual is not able to just reproduce by itself. In sexual reproduction, it takes two parents to actually produce the offspring. The other type of reproduction is called asexual reproduction, and that is how cells reproduce. Cells only require one parent to produce offspring. In prokaryotic cells, this is a very simple process. A prokaryotic cell, the only thing it needs to do is it needs to grow a little bit larger and replicate its DNA. Once it grows larger and has replicated its DNA, the prokaryotic cell can divide into two new cells, and there we go. That is the life cycle of a prokaryotic organism. All they need to do is get a little larger and replicate their DNA. For a eukaryotic cell, this process is a little bit more intense. So eukaryotes, they have two main stages of their life. You have interphase and you have the mitotic or the M phase. Interphase contains three different phases within it. Those are G1, S, and G2. And we will talk about what those are in a few minutes, as well as what happens during mitosis and cytokinesis for the mitotic phase. So let's look at these individually. How does that cell cycle actually look? And here it is. First, interphase and M phase. Interphase is the vast majority of a cell's life, and M phase is just that little bit at the top. So the vast majority of the time that a cell is alive, it will be in this phase called interphase. And interphase, as you saw, is broken into those three different parts. G1, S, and G2. And a cell will typically go through this cycle in a nice orderly fashion. if a cell is not supposed to go through the cell cycle. This is due to some sort of level of regulation. For example, we happen to be multicellular organisms. We don't need all of our cells to reproduce all of the time. Yes, we need to have our cells grow and develop and reproduce when we need to heal damaged tissue or when we need ourselves to grow. But if the cells are not supposed to reproduce at any specific time, if they're supposed to maintain the number of cells that they happen to have, then those cells will actually exit, in a sense, the cell cycle, and they will go into this little section, which we call gap 0. Once a cell enters into gap 0, it can enter back into gap 1 if it's supposed to, but it might just stay into gap 0 for the rest of its life. A lot of nerve cells, when they enter into gap 0, they never exit gap 0. They stay there for the entirety of our life. Heart cells do the same thing. Those cells multiply to a specific number of cells, and then they all enter into gap 0, never to reproduce again for the rest of the organism's life. But some cells, like your liver cells, they will enter into gap 0. But then, as the liver gets damaged, the remaining cells can exit gap 0 and enter back into gap 1 so they can go through the cycle again to reproduce, to repair the damaged liver. That's how cells work through the cell cycle. The purpose of each phase is very straightforward. So G1, that actually stands for gap 1. And that is where the cell is just going to perform its general, normal living functions. That means it's going to be responding to stimuli. That means it's going to grow, maintaining homeostasis. It is going to be using up energy, and it's going to have to take in food. During gap 1 of a cell's life, the cell is going to be undergoing many of the different processes that we've already gone over, utilizing the endomembrane system, utilizing the mitochondria. If it's a plant cell, it's going to be utilizing the chloroplast. All of the organelles working together to maintain the cell's life. Then the cell can progress onto the S phase, where the DNA inside the nucleus will be replicated. Before this point, the cell only has one copy of its DNA. And if the cell were to reproduce, then neither of its offspring would end up with the full amount of DNA that the cell is supposed to have. So this stage is crucial for a cell to actually be able to reproduce. Without replicating the DNA, the cell would be unable to produce daughter cells. And that takes us to gap 2, where the cell will actually check to make sure that those duplicated chromosomes are all duplicated correctly. And then it will also do the rest of the cell's normal functions. And then the cell will enter into the M phase, which is where the DNA is going to be separated, sorted, packaged into new nuclei, which would be mitosis. And then in cytokinesis, that is where the cell will divide and become 2 new daughter cells. The original cell ceasing to exist as it divides and becomes the 2 daughter cells. But to understand all of what we're going to be talking about as we go through the cell cycle and as we go through specifically the S phase and the M phase, we really have to understand some vocab. So first vocab is a chromosome. And we did go over this before, but I just want us to reiterate, a chromosome is a strand of DNA. Chromatin, that is the form that the chromosomes will take during interphase. The chromosomes during interphase are not condensed how we normally think of them. They're this unraveled form, and that means that they are going to be in the form of chromatin. When we're going through the mitotic phase, M phase, we'll talk about replicated chromosomes. That's where we have 2 strands of DNA that are exact copies of each other that happen to be attached at this junction called a centromere. Replicated chromosomes are what the S phase generates. The S phase is about replicating those chromosomes, but they don't condense until the M phase. They stay as chromatin during the entirety of the S phase. And then a chromatid, that is one of the copies that exists on a replicated chromosome. So when you have two copies that are connected together, each copy is called a sister chromatid. So again, chromatin, that's unraveled chromosomes, that's unraveled DNA. It is not condensed. It is a decondensed form. It has to be in this decondensed form so that the cell can use it. When the chromosomes condense, that makes the DNA inaccessible and unusable. We must remember chromosomes, they are made up of these things called genes, and each chromosome has specific genes that make it up. This would be an unreplicated chromosome. A replicated chromosome would appear like this, where you now have two copies. Each copy is called a sister chromatid, and they're connected right here at the centromere. Okay, so that would be a replicated chromosome. The only time that you would see a chromosome condensed is during the M phase. The entirety of interphase, again, is just going to be chromatin. So there is no such thing as a condensed chromosome except for in the M phase. So in summary of all of this, well, all cells are going to grow and develop, and they're going to go through that process called the cell cycle. To reproduce, these eukaryotic cells are going to have to go through gap 1S, gap 2, and then they're going to have to go through the M phase. And the majority of a cell's life is going to be in the phase that we call interphase, where it's growing, developing, maintaining homeostasis, responding to stimuli. All of those processes that make its life exist are going to be taking place during interphase. In our next lesson, we'll talk about one part of interphase. It's the S phase. We're going to focus in on how the cell replicates its DNA. Until then, be awesome, stay awesome.